So our next speaker is Marge Daughtrey from Cornell University. She is going to be covering the fundamentals of biological control of fungal and bacterial diseases. So before we get started, again, I just want to thank Syngenta for their sponsorship of this webinar. If you don't have the handouts for Margery's or Matthew's presentation so far today, go to the materials section of the Go to Training Control panel and download those and any of the other PowerPoint PDFs that you want to have for your records and for note taking. But without any further delay, I'm going to hand this over to Margery. So Margery, hang on while I send this over to you. And let me turn your microphone on. Okay, Margery, you should have sound and you have the computer as well.
Okay, I assume you can hear me.
I can hear you, yes.
All right. Okay, and I believe I have the presentation up, so we should be set to go. Hello, everyone. I've been enjoying listening to everything there is to know about biocontrol of insects in the greenhouse, and we're switching to another whole kind of topic now with my presentation and Matt's, because we're going to be talking about biocontrol in the disease arena, and hopefully you'll find this interesting and helpful as well. The good thing is that biological control of diseases is actually considered to work much better in the greenhouse than it does out of doors. So those of you who are concerned with protecting your greenhouse crops may find that you can be some of the first people to get good at doing biological control of plant diseases. The basic idea here is that biological control is pitting one microbe against another. That's the first thing that's unique about this, is that you're working with two invisible things. It takes not just imagination, but skill to work in this area. But some of the same concepts are involved as in the insect control arena. You're not going after controlling mushrooms that are perfectly visible. You're actually working against the hyphae, the filaments of the fungi that might be down in the soil or in the container mixes that you're dealing with in the greenhouse. These are the strands that would be gaining access to the plant's root systems and then penetrating and causing root rot or crown rot in many cases. And then there are also biological control strategies designed to work on the upper parts of the plant, on the foliage or on the flowers. And in this instance, they're most often trying to block the successful invasion by spores. So stopping the normal germination penetration of a spore trying to gain access to the plant is the goal of the biological control. Here is an array of symptoms of the rust diseases, which are one of the examples of the kinds of diseases that might be assailing the top part of the plant. You see here 3 different kinds of rust. And at the bottom of the slide are spores of the chrysanthemum brown rust, 2 different kinds of spore, as these would land on the plant surface. If they were to encounter an unfriendly biological control organism, the infection might be prevented. The other thing that biocontrols can do in the disease category is to go after bacteria in some cases. And as you know, we're not very good at controlling bacteria with chemicals, so we may actually find out that we can do a better job with biologicals. It wouldn't take much to do a better job. The bacteria, of course, are actually, when you don't have very many of them, quite microscopic creatures. The little tiny dots here on this Petri plate would represent millions of bacterial cells that have resulted from a single cell dividing into 2, and those 2 dividing into 2 each, and progressing till you have something that's visible to our eye. The biocontrol is going on at the level of individual bacterial cells interacting with individual strands of fungi or other bacterial cells. The kinds of bacterial diseases that we might be able to fight back against with biocontrol methods would include things like a Xanthomonas leaf spot on English ivy, which is a common problem in the greenhouse, or perhaps a Pseudomonas leaf spot that would occur on chrysanthemums under wet and warm conditions, or perhaps the bacterial leaf spot on zinnias, which is a very common xanthomonas disease that is most problematic when people are trying to grow potted plant zinnias towards the end of the bedding plant season. If you have a vascular wilt disease, such as those caused by Ralstonia solanacearum, you probably are not going to see much benefit from a biocontrol kind of an approach. And that's because a vascular wilt, such as a fusarium wilt or a verticillium wilt or a Ralstonia, represent diseases where the action is inside the plant, and the biocontrols that we're working with operate more in the environment on the surface of the leaf or the environment around the root system called the rhizosphere, and they're not penetrating into the plant to interact with pathogens there. So think of the biocontrols as being helpful for dealing with non-systemic disease problems. Here you can see a close-up of a geranium stem that's been cut and the bacteria of Ralstonia oozing out from the xylem. We don't have a way of injecting plants with biocontrols to fight back against diseases. The basic concept of biocontrol for disease is that you're sending your team in ahead of time to protect against the fungi and the bacteria that might cause diseases. You're sending in your guys against the fungi or the bacteria. One of the big differences in biocontrol for diseases is that the materials that are used for biological control for disease are regulated by EPA. This isn't true for the biocontrol of insects. And EPA has its own unique definitions for things, including a definition of what they consider biopesticides, which includes some things that are not living. So for the EPA type of definition, a biopesticide is something derived from natural materials, which includes microbes like fungi and bacteria, animals and plants, and even some of the minerals. Under their category of microbial pesticides, you'll find the beneficial bacteria and fungi, the main things that we'll be talking about here today. But also under EPA's biopesticide definition, things called plant-incorporated bioprotectants are included, and this would even include genetically modified organisms. So there's an interesting shift of concept there when you consider those as potential biopesticides. And then, of course, when you're dealing with plant diseases, you don't have the ability to do things to trick the pathogens, like the use of pheromones for control of some of the insect problems. So we don't really have any of the biochemical pesticides in that category of EPA's definitions. One of the most important things for dealing with insect control with biologicals is certainly to get them there early. And the same concept definitely applies with disease management. You want to use the biocontrols preventively, and you want to make really sure that they are not being put in to control a situation that is already out of control. It's nothing like using a chemical. Chemicals can often allow you to come in after a problem has gotten started and to at least halt them in their tracks at that point and throw away the victims. With biological control for diseases, you want to have your beneficial organisms there in advance. You also have a great deal of difference in this system than using parasites and parasitoids. I think the main difference is that you can't see them. So you're going to be working with things that you know are there, but you will not be able to watch the battle between your microbe and the disease-causing microbes. So how do these plant disease biocontrols work? They have a number of different modes of action. The first one is just occupying a niche. Essentially, the fancy term would be competitive exclusion, and the idea is that if there is a zone along the root system of your plant, if you have occupied that zone with a beneficial organism rather than a disease organism, it may physically keep your disease organism from getting contact with the root and getting established. So just physically being there is one of the things that these organisms can do. A second concept is predation. That sounds a lot like the insect world, but in this context, it involves something like a Trichoderma fungus being able to bore into a Rhizoctonia hypha and literally grow inside the Rhizoctonia and feed on it from within. There can be very direct predation in some of these cases. There can also be antagonistic metabolites, which is a fancy way of saying that the biocontrol organisms are very good competitive microorganisms. And the way they express their competition is often by producing chemicals that help them and theirs to survive and sort of depress the populations or the success of other nearby organisms. One of the most common groups of chemicals in this category would be considered antibiotics, which we're familiar with because we take antibiotics. Nutrient competition can also be involved with these biocontrol organisms. You can have, in particular, competition for iron is very important among the different bacteria around the root system and the fungi that live in that area. So if you have a biocontrol that sucks up all the iron, it might make it impossible for some of the disease organisms to function well in that altered environment. Also, there are some interesting factors of stimulating the defense of the plant. And these are natural systems that we are learning more and more about how to manipulate. There are concepts called induced systemic resistance and systemic acquired resistance, which are involved in making a plant naturally sensitized so that it will be perhaps less likely to grow rapidly, but more likely to be able to defend itself against disease organisms. There's a shift of its metabolism to defense away from active growth. As long as there isn't too much price to pay there, the increased defense can be a very positive thing for a plant that is led to that by organisms interacting with its leaves or its roots. The good thing about all this is that biological control can be very complex. Most of the agents that we are dealing with use a couple of these mechanisms at least, if not all of them. And because they have a multifaceted approach to doing biocontrol, that means that there is very little likelihood of resistance development to this kind of a control mechanism. Anytime you don't have a single mechanism for the control, the way you have with the newer systemic fungicides, you have a much better chance of a lasting effectiveness from the biocontrol that you're dealing with. There are two kinds of biocontrol organisms that are most common out there at this point in time. Some of them are fungi, such as the Trichoderma species, and some are bacteria, and the two genera that are primarily involved there are the Bacillus species and the Streptomyces species. Streptomyces is an actinomycete, and at times in the past that was considered a fungus, but now we're considering it a form of bacteria. Getting back to our microbe versus microbe idea, there are certain fortes to the different bioantagonists. The Trichodermas work best against other fungi. The Streptomyces seem to work best against fungi. And Bacillus is gaining a reputation for being particularly useful against both bacteria and fungi. Let's think a little bit about the prey for the biocontrols, the things that you're actually trying to prevent. All of the biocontrol organisms that are available on the market today seem to be especially good against powdery mildew, and that's a very good thing because powdery mildew is a very conspicuous disease that we need to keep under control in the greenhouses. There are also a wide number of products available now. We have two products that are Streptomyces based, Micostop and Actinovate. We have three listed here that are Bacillus products based on different species of Bacillus, cease, companion, and double nickel. And there are likewise a number of plants that are used in drench applications or enclosed in mixes to help to retard disease problems that would affect primarily the roots or the crowns of plants. And this includes the clumps that you see here, the Trichoderma organisms found in Root Shield and Biotam, also the Streptomyces materials found in Micostop and Actinovate, and then the Bacillus materials found in Cease and Companion and Double Nickel, and also in a mix, the ProMix with biofungicide. One of the things you'll notice that on this listing, there are a number of designations here at the end of the Latin name, things like K61 here for the Streptomyces griseoviridis. That's an indication of a particular strain, And you should always be aware that a lot of work went into developing each of these organisms. Each one of them that is marketed in this fashion has really withstood all the comparisons against others of the same genus and species and has proven to be long-lasting and effective compared to others of the same type. So there's a lot of selection that's gone into each of these. Let's think about how you would tackle the soil-borne problems. You would be interested in using biocontrols to work against Rhizoctonia solani, which causes a lot of root rots. You would be interested in controlling things like Phytophtheras, which have that nice vulnerable zoospore stage in the soil that could easily be competed against by biocontrols. We see a lot of root problems in crops like lavenders from Phytophthora, and it would be tremendous to get them protected from the beginning so that we didn't have as much loss in those. We have seen lots of problems with Calibrachoas having Phalaeviopsis difficulties, and ordinarily we're using chemical control to fight back against Phalaeviopsis, but this is an experiment from a few years ago where we saw some benefit from some of the biocontrol treatment. If you look at the left, non-inoculated plants had this size to them at the end of the experiment. The inoculated controls that were inoculated with the Phalaeviopsis were much smaller and sickly. And over here you can see that the root shield gave not as good protection as the thiophenate methyl in this bar, but as good as TerraGuard and Allude. One of the options is to work with a mix from the beginning that has mycorrhizae in it, as well as Bacillus pumilus, and this is offered as one of the ProMix formulations. There are also in that mix there is the opportunity to have induced systemic resistance coming from that bacillus. So there could be a lot of interesting benefits from working with that kind of material, and you might find it would be helpful to also supplement with one of the drench materials that provides biocontrol. We're also interested in controlling Pythium when we're using biocontrols. Here's an illustration of an experiment where the first row shows you plants that were not inoculated. The second row shows you plants that were inoculated with Pythium, and they're all growing very small. Here you have 4 rows that were all treated with different biocontrol treatments. And you can see a range from dead plants to ones that look just as good as the controls. Over at the right, you see plants that were drenched with chemicals. And this is a typical kind of a scenario in experiments that we run because we're not able to give an inoculum load that's similar to real life. We do inoculate fairly heavily in our trials, and I think the irregularity here is probably going to translate to much better success in the real world situations. There are particular tools available for biocontrol in greenhouses. We have, and this looks like a law firm, Harzianum, Hamadum, Virens, Asperellum, and Gamzii. These are the Trichoderma species that can be used. We also have here the best known of the biocontrol agents. Root shield is used as a common drench to protect plants against various fungi in the soil, things like Pythium, Rhizoc, Fusarium, Phalaeviopsis, and Cylindrocladium. And there is a newer product that has a combination of Trichoderma harzianum and Trichoderma virens that goes by the name of Root Shield Plus that also suppresses Pythium aphanodermatum and gives improved benefit against Phytophthora. There's a Biotam product that also is a combination that has Trichoderma asperellum and Trichoderma gamzii that's effective against roughly the same kinds of soil organisms. This one has a funny quirk in that it has a label warning because it can pose a risk to beneficial beetles. So that's something to keep in mind if you're doing some of your insect biocontrol with beetles. Streptomyces species are available that sound a lot like an antibiotic you might take. Grisia viridis is one of the ones that's found in the material Micostop, and it can be used against a number of soil diseases and also helps suppress botrytis when used on foliage. Then you have Streptomyces lydicus. That is useful against, again, a number of the soil organisms as well as powdery mildew and downy mildew above ground. We've used it on powdery mildew of poinsettia, for example, and found that it can do some really useful things there. If plants are not treated, the disease can progress in this fashion. The activate with Latron, it can provide sort of a medium level of control compared to some of the systemic fungicides that are down here at the bottom. Bacillus species, another one of the options. You have Bacillus amyloliquefaciens that's found in double nickel, which also has foliar as well as root disease problems that it addresses. Then you have Bacillus subtilis, again, one of the better known of the biocontrols, found in companion and cease. And the companion material has one outdoor product for turf and landscape use and one that's used indoors for greenhouse and nursery ornamentals. Again, going after the standard root fungi as well as the foliar diseases. Cease has Bacillus subtilis within it, and it's one of the products that has an OMRI listing, and that's also a big advantage. You can use a lot of these products for biocontrol. Botrytis is one of the things that's often addressed by these materials. Here's a little bit of data from an experiment we did a few years ago where the cease material gave as good control as Decree, one of the standard fungicides for botrytis management. And then here is some data from Jean Woodward on use of Rhapsody, which has the same active ingredient as cease, where it did as well as Eagle and Compass in reducing the powdery mildew on hydrangea. We've also seen good effects using these biocontrols in alternation with other products. Here you have untreated plants with a large amount, almost 80% coverage with mildew, some depression of that disease by an actinovate treatment, some depression of that disease with a Rhapsody, which would be like a cease treatment today, and then extremely good results if you're alternating the products that are biocontrols with things like cease or actinovate. And that's, I think, one of the most useful approaches to biocontrol for beginners is to alternate between chemical and bacterial materials. Looks like I'm running out of time here, so I would refer everyone to the fact sheet that was made available to get a little bit more information. And I know Matt's going to be able to clean up anything that I haven't addressed yet. So I'd like to stop here and ask for any questions.
Okay, Margery. Let me look here. What do you think about tank mixing conventional fungicides with biorationals?
I think that tank mixing conventional and biorational is fine, but I just as soon alternate. One of the things that seems to be problematic is the cost of doing the tank mixes. So I think particularly with these kinds of products, which are generally used every 7 days rather than further apart, it's going to be a good strategy to alternate rather than to mix. So either with a biorational or with a biological, I like the idea of alternation at this point.
Okay. And do you see better control of root disease with Trichoderma or Streptomyces?
I've not actually pitted the two against one another. I've tended to work on one at a time, trying to help develop the efficacy of the products. I've seen some efficacy from each one of them. So I think each grower has to do the same kinds of experiments because they have to work on with their medium and with their crops. So I would encourage anyone who wants to start out with biocontrols to begin by leaving some untreated plants as they treat the bulk of their crop and see how things do. I think that's the best way for them to learn what's good on their crops.
Okay, how about does Regalia help prevent downy mildew on greenhouse herbs?
Oh, I wish Regalia would help prevent downy mildew on greenhouse herbs. I do think that in particular the downy mildew on basil is a horrendously difficult disease to manage, and I think it is a bigger problem than what Regalia would be able to deal with. I didn't mention the Regalia yet. It's at the tail end of the presentation. That's in the not the strictly biological category. It's a weed extract that is going to definitely help with powdery mildew, probably help a little bit with downy mildew, but I think you're going to need something much more potent to really cope with downy mildew on greenhouse herbs, such as a phosphorus acid. It's a very difficult disease to cope with because it's like the Ralstonia and the Fusarium. It's a systemic disease. So you need a really careful program to manage that one.
Okay, does anybody else have any questions for Margery? Let's see, are there any antagonistic responses seen when using several of these biorationals in a row or even in tank mixes? I think that's the question that they're asking.
That's a very interesting question. I've never looked into that. I think Matt would probably be able to address that during his presentation because he's probably been in the world where that's coming up. Within the BioWorks line, for example, they're dealing with cease application as well as root shield application. So if there were either synergism or a problem with the two together, they probably do know that. So let's ask him that question when he comes up.
Okay, I think a couple people are having some issues with the sound when you're replying to the question about the downy mildew on basil. Could you repeat that really, just summarize that really quick for us, Margery?
Sure, happy to. The downy mildew on basil is a systemic disease, and it's very hard to control. It also can start from infected seed, so the plant can begin with it before you've ever started trying to control the problem. So I don't think it's a good candidate for biological control. I don't know of any that would have a really significant effect on it. Seems like phosphorus acid compounds are going to be much more relevant to keeping it from expressing itself in the greenhouse.
Okay, I think that's all the time we have now. If growers have questions that didn't get answered, Margery, is it okay to email you?
Sure, that's fine.
All right, nothing like putting you on the spot and
No,
making you answer in a positive way, right?
Right.
All right, thank you very much, Margery. I appreciate it, your time that you gave us today and all the information that you provided.